All right, everyone, please confirm me. Am I audible to everyone? Everyone, please confirm me. Am I audible to everyone? Everyone, please confirm me. Am I audible to everyone or not? All are very silent. No one is speaking. All right, come on. Please be active. Okay, don't be dull in the class. All right. Okay, not a problem. Okay, now let's please continue, and every one of you please try to understand every concept very correctly and keep answering my every single question. All right, so in the last class we have completed the prokaryotic DNA replication and we have started discussing about the eukaryotic DNA replication. I'll quickly summarize everything in this span of 1 minute. Just try to understand. In case of a prokaryotic organism, how many ORCs are there? Only one ORC is there. Tell me, Manju, in case of a eukaryotic organism, how many are there? I have very clearly told you, in the eukaryotic organism we generally call it as the what? Replicator. I believe every one of you, there are more than 150 replicators are present, correct na? Sorry, there are more than 400 replicators are generally present. I believe every one of you remember, in case of a eukaryotic organism we have taken yeast as the best example, correct na? And in the yeast, this particular replicator also called as what? ARS, autonomously replicating sequence. I believe every one of you remembered it very clearly. Okay, now I believe you all people remember my statements very correctly. Now you all people has to answer the question correctly. The eukaryotic DNA replication is always associated with the cell cycle. If you quickly recall G1 phase, S phase, Z2 phase, as well as the M phase. However, we say G1, what we generally say that G1 phase is the preparatory phase for the S phase. Similarly, we use it to say that Z2 phase is the preparatory phase for the M phase. Whenever we use the word G1 phase is the preparatory phase for the S phase, what does it mean, friends? I believe you all people remember all the enzymes required for the DNA replication are getting synthesized in the G1 phase, but they are not at all becoming active in the S phase. Sorry, not active in the G1 phase. When they become active in the S phase only. Similarly, however, we call G1 phase is the preparatory phase for the S phase. Similarly, friends, we call Z2 phase is the preparatory phase for the M phase. There is no point of getting confused over there. I believe every student remember. I have clearly explained it to all of you. First step in the DNA replication, all the students listen. What I told to all of you, in the G1 phase, a pre-replicative complex will be formed. Friends, what is pre-replicative complex formation? Up to helicase. But helicase never ever be active. The pre-replicative complex converting into the active replication complex in which phase? We very clearly studied that it is converting into the active replication complex in the S phase. Now every single student please answer my question. MCM having how many subunits? Every student please answer my question. MCM, which is a MCM protein which is acting like a helicase, having total number of subunits. How many number of subunits are present in the MCM protein? Very good. I hope every student remember MCM, what I told you, Parul. Very good. MCM is generally hexameric helicase protein. Correct or not? Why everyone forgot it? It is generally having how many subunits? 6 subunits are there. Generally. In some proteins, it is having 6 to 10 subunits also. I have told to all of you, even in the prokaryotic DNA replication also. Everyone forgot it and no one is answering except one or two. Very clearly I told to all of you, right? Very good. MCM, correct. Hexameric helicase. Correct, na? I believe every student remember this particular picture very clearly and keep answering my questions. So let's consider this is the replicator region. So to this replicator region, who will go and associated first? First ORC will go and associated. That is the name of the protein itself, friends, origin recognition complex. Origin recognition complex. Now the origin recognition complex first recognize the replicator. I believe all the students remember immediately what is going to happen. This ORC, listen everyone, it is going to recruit either CDC6 or CDC18. Don't get confused. Then CDC6 or 18 will come and attaches. Then immediately there is a recruitment of the CDT1 protein. So what is happening now? ORC, CDC6 or 18 and CDT1. These three together recruiting the MCM. You all people can see the picture very clearly. MCM is a hexameric subunit. It will come and attaches. Now tell me all the students, please answer my question. Every student, everyone answered, okay? Don't be silent. Until this MCM recruitment, where it happened? In the G1 phase. Now my question to all the students is very clear. Why the DNA replication is not happening in the G1 phase even though MCM is present? Even though MCM is present, why the DNA replication is not happening in the G1 phase? What is the answer? Every one of you. Why DNA replication is not happening in the G1 phase even though MCM is present? The answer is very clear. MCM is not active. Whenever the MCM become active, then only it start doing the DNA replication. I don't know why people are not answering. Everyone is listening like robots. There's no use. Okay. There are how many subunits are there in the MCM? There are 6 subunits are there in the MCM. Out of the 6 subunits, every student listen. Minimum, I am using the word minimum. Minimum 5 subunits. Minimum, correct. Minimum 5 subunits has to be phosphorylated. That is an important statement here. Correct, na? Minimum 5 subunits has to be phosphorylated. Very good. All of your answers are very correct only. Now the question is that how it is getting phosphorylated? Who is doing the phosphorylation? That is important for the exam. If the MCM wants to become active in the S phase, MCM has to be phosphorylated compulsory. But who is doing that phosphorylation? That phosphorylation can be done by important proteins called as CDK and DDK. They are extremely important. I want all the students has to remember compulsory for the exam. CDK and DDK, what they are doing, friends? They are phosphorylating. No point of getting confused. They are phosphorylating 5 of the 6 MCM subunits. 5 of the 6 MCM subunits, they are phosphorylating. Then only they can act like a helicase. No point of getting confused over there. Now all the students understand this particular diagram, friends. This diagram and this diagram both are extremely important diagrams. Okay. There is a possibility that you gonna expect the question in the graphical form, which we already got it. Even in your 2024 July paper also the question has come in the part B section. One second. First you answer this question. All the students answer this question. This is question number ID 42 in the morning shift. Part B question. The question paper is moderate. It is very doable, but most of the students, because of overconfidence and some students failed in analyzing the questions, you could answer very easily. Anyway, not a problem. Let's see that. Everyone quickly. Parul, what is the answer? Divya? Anyway, the question is so simple, right? In eukaryotic cells, DNA replication is restricted to the S phase of the cell cycle because, why? Because MCM is getting phosphorylated only in the S phase. So option 1 says that DNA polymerase is present only in the S phase of the cell cycle. This is the wrong statement. DNA polymerase is present in the G1 phase also. ORC is a name of the protein. Recognizes only origin in the S phase. This is wrong statement, right? Recognition also happen in the G1 phase only. MCM helicase get activated in the S phase of the cell cycle, right? MCM helicase get activated in the G1 phase of the cell cycle. That is wrong statement. The correct answer will be the number 3. No point of confusion at all. In previous year, they have asked the question in the form of a graph. Okay, that too in the 4-marker question. Anyway, let me show you. Okay, try to understand the concept very correctly, friends. You people can observe this picture very clearly. I am summarizing here everything. This is what protein? ORC protein. Then followed by CDC6R18, CDC6R18. Then this is the CDT1 protein. All together recruited what? Very good. All together recruited the MCM complex. But where it happened? It happened only in the G1 phase. Then immediately in the S phase, what is happening? In the S phase, exclusively the level of the CDK. That is an important statement to be remembered. The level of the CDK is extremely higher in the S phase. That is the reason CDK will come and do the phosphorylation of the 5 subunits very easily. Whenever it do the phosphorylation of the 5 subunits, then only MCM act like a helicase and it starts opening the double-stranded DNA very easily. Now this is the most important picture that I want all the students has to understand my statements. Let's quickly compare with the prokaryotic organism. In the prokaryotic DNA replication, what generally happens, friends? RNA primase is the enzyme which will come and add the primer. Whenever it will come and add the primer, immediately what is going to happen? Tell me. Then DNA polymerase enzyme will come and elongate it. But we studied very clearly, in case of a eukaryotic DNA replication, friends, there is nothing called as RNA primase. There is no enzyme called as RNA primase. Now, every student, please answer my question. Tell me, Manju, tell me, every one of you, what is the name of the enzyme that can add the primer in the eukaryotic DNA replication? All the students. What is the name of the enzyme that will add the primer in the eukaryotic DNA replication? All of you. I believe all of you remember DNA polymerase alpha. We already studied it. What is the speciality of the DNA polymerase alpha? It is having the primase activity also. At the same time, it is having the polymerase activity also. In the next time, there is a possibility of asking this kind of question. Which of the following enzyme having both primase and polymerase activity? Which is the DNA polymerase alpha. Every student, pay attention towards the diagram here. You people can observe. DNA polymerase alpha, can you see that? It added the primer. DNA polymerase alpha, it added the primer. There's no doubt about it. After adding the primer, the polymerase alpha itself elongating the new strand also. You can see that. After addition of that, DNA polymerase alpha itself adding the nucleotide also. I believe you all people remember. Anyway, it cannot continue not more than 20 to 50 nucleotides. So what is the reason why it cannot continue? The only reason is that it is having extremely low processivity and it do not have any DNA proofreading activity. It do not have the proofreading activity. At the same time, it cannot do the, it is having very low processivity. That's the reason later DNA polymerase alpha can be replaced by the delta. There is no point of getting confused over there, and it is easy to understand also. All right, so same thing. I believe all the students remember the concept of the regulation in case of a prokaryotic organism. I believe you all remember. What is the mechanism of the prokaryotic DNA replication regulation is called as? I believe you all remember. Sequestration. That is discriminating between the daughter strand and the parent strand. I believe you all people remember that. Now, if you look into the regulation of initiation in the eukaryotic DNA replication, what I told to all the students, everything is under the control of the CDK molecule. And friends, what is the CDK molecule? No point of getting confused. CDK molecule is the one which is generally involved in the activation of the MCM. That is the helicase. CDK and DDK are what? Kinases. What is the function of a kinase? Doing the phosphorylation to the MCM protein. No point of getting confused over there. Whenever, all the students understand, use the brain. Whenever the CDK activity is very low, whenever CDK activity is very low, because friends, this only they will give in the form of a graph. When the CDK activity is low, all the students understand, pre-replication complex will form. But pre-replication complex never be active. I hope every student is getting this, getting the meaning of this line. When the CDK activity is low, when the CDK activity is very low, actually it is very low in the G1 phase. Now you only tell me, good morning. Actually, what happens in the G1 phase? In the G1 phase, undoubtedly, friends, pre-replicative complex will form. No doubt about it. But the point is very clear. Pre-replicative complex never ever become active. Never ever become active. Now you tell me, what happens when the CDK activity is higher? You all know very well. Where the CDK activity become higher, generally CDK activity will become higher in the S phase. In the S phase, what is happening? Understand, my friends, whatever the pre-replicative complex that is already formed, I want you to understand my statements correctly. Whatever the pre-replicative complex that is already formed, that becomes active, which means that MCM is getting activated, entire replicative complex becomes active, then the replication will start. But what you have to understand, no new pre-replication ever be formed. That is an important statement. No new pre-replication complex never be formed, but whatever the replication complex that is already formed, it becomes active. So these are the differences that they give in the form of a graph. So you people can observe here very clearly, very easy to understand also. Very good. When the CDK level is very low in the G1 phase, when the CDK level is very low in the G1 phase, what is going to happen, friends? Pre-replication complex will form, but the pre-replication complex never be active. Point is very clear. Then here what is going to happen? In case of a S phase, CDK level is higher, which means that pre-replication complex converted into active replication complex. This point is very clear. And second point is that no new pre-replication complex never be formed. That's all. Simple difference. New pre-replication complex never be formed. Whatever the pre-replication complex is already formed, that becomes active. There is no point of getting confused over there. I want you to compulsory remember for the exam. So in one single line, what I want to tell to all of you is, entire eukaryotic DNA replication is under the control of CDK protein. This is what I want to tell you. The entire eukaryotic DNA replication regulation is under the control of the CDK, where the level of the CDK is very low in the G1 phase, where the level of the CDK is extremely higher in the S phase. There is no point of getting confused over there, and it is easy to understand. Now I want to tell you one more example here. There is, like, however CDK is exclusively important in the humans also, similarly in case of a metazoan cells, instead of the CDK there is another protein which is called as a geminin protein. So sometimes they will ask this kind of question, so you have to know it. Just remember the name enough. Just remember the name enough, because the function performed by the geminin and the function performed by the CDK, which is almost similar. The only difference here is that the geminin protein exclusively present only in the metazoan cells. No point of getting confused over there. See, it's not only about understanding, friends, it is all about applying your knowledge. So try to get the concept in a right way, okay? Then you can answer the given question so easily. Next one, the process of the elongation. Let's try to understand the process of the elongation. I have very clearly told you, whether it is a prokaryotic organism, whether it is a eukaryotic organism, doesn't matter. What happens generally, whether it's a prokaryotic or eukaryotic organism, in case of a elongation, only addition of nucleotides will happen, nothing else. Addition of nucleotides. I believe all the students remember, in case of a prokaryotic organism, there is an important enzyme called as DNA polymerase 3. That is the one which generally synthesizes the leading strand also. That is the only one which synthesizes the lagging strand also. Very important, friends. Now I want you to answer my questions very correctly. In case of eukaryotic organism, DNA polymerase delta is the one will synthesize the leading strand. Sometimes lagging also synthesized by the polymerase delta only, friends, no doubt about it. But sometimes when it comes to the lagging strand, polymerase delta replaced by what? Epsilon. Polymerase delta is replaced by the epsilon. No point of getting confused over there. So understand the points correctly. The leading strand is the template strand that is being replicated in the same direction as that of the replication fork. What is that? Polymerase delta. I believe, friends, what we all people discussed, polymerase delta cannot do the DNA replication in the starting. Who will start the DNA replication? Tell me, all the students. Polymerase alpha. Let me write here. This is one template. This is another template. Now what the polymerase alpha is doing? First it is adding the primer. Correct na? After addition of the primer, alpha itself continuously adding the nucleotides. Correct or not? Alpha itself adding the nucleotides continuously up to 20 to 50. But since polymerase alpha having the lowest processivity, then what is happening? Alpha can be replaced by the delta. Now question is very clear, friends. Who is going to replace the alpha with the delta? Can any student please answer me? What is the protein that replaces the polymerase alpha with delta? Can any student please answer me? What is the enzyme that replaces the polymerase alpha with delta? Everyone. Come on. I hope we all people remember, friends. In case of a prokaryotic organism, we have a beta clamp. But tell me, Parul, in case of a eukaryotic organism, we don't have a beta clamp, right? Then who is doing the function of the beta clamp? I forgot everyone. PCNA. Proliferating cell nuclear antigen. It is the protein which is replacing the alpha with delta. Correct or not, everyone? Then only delta continuously starts synthesizing the daughter strand. No point of getting a very good. Correct, Ashoda. So no point of getting confused over there. That is what strand synthesis, friends? That is the leading strand synthesis. The point is so clear. Then What is very good, very good, correct. What is the lagging strand synthesis? We discussed lagging strand synthesis is always discontinuous synthesis. What is the meaning of a discontinuous synthesis, friends? So it is the daughter strand is synthesizing away from the replication fork, correct or not? Suppose this is the direction where the replication fork is moving, but if you look into the lagging strand, it is coming opposite way, correct na? That's the reason we call it as the discontinuous synthesis. And mostly lagging strand synthesis exclusively mediated by the polymerase epsilon. The lagging strand synthesis exclusively mediated by what, friends? It is exclusively mediated by the polymerase epsilon. There is no point of getting confused here, friends, and it is very easy to understand also. Now, what is the length of the one Okazaki fragment? I believe every student remember, in case of a prokaryotic organism, what is the length of one Okazaki fragment? I believe you all people remember. In case of a prokaryotic organism, one Okazaki fragment, it is 1000 to 2000 nucleotides, correct na? But total how many are present? Total few thousands are present, that's all, few thousands. But if you observe the eukaryotic organism, friends, one length of one Okazaki fragment, only one length of one Okazaki fragment is 100 to 200 nucleotides, that's all. But how many number they are present, friends? They are generally present in millions in number. There is no point of getting confused over there. What, friends? They are generally present in the millions and millions in number. Easy to understand this particular point. Okay, last and final question I want every one of you to answer. Everyone answer. Don't be silent in the class. Tell me, all the students, what is the name of the enzyme? Tell me, Parul, correctly, that removed the primer. That removed the primer in prokaryotic organisms. Everyone, what is the, tell me, Vijay Lakshmi, what is the name of the enzyme that removed the primer in a prokaryotic organisms? What is the name of the enzyme that removed the primer in a prokaryotic organisms? Tell me, Divya. What is the name of the primer that removed the, name of the enzyme that removed the prokaryotic organism? Removed the primer in prokaryotic organisms. No one remember it. Huh? DNA polymerase 1. Correct or not? DNA polymerase 1, we also call it as RNase enzyme. No one is answering. Everyone forgot. Huh? Everyone is just listening the lessons and leaving it. No one is reading. DNA polymerase 1 or RNase is the enzyme that remove the primer, and I believe you all people remember that will only replace the nucleotides also. Correct or not? Very good. But friends, when you look into the eukaryotic organisms, what is the name of the enzyme that is removing all this particular primers? All the students remember, all the primers can be removed. Very important, friends. Correct na? All the primers can be removed by an important enzyme called as RNase H1. Correct na? RNase H1. No point of confusing. Except last primer. This is a very important statement. This is also direct question which we got in the exam. In case, because you have to compare between the prokaryotic and eukaryotes, then it is easy to understand the concept. In case of prokaryotic organism, as you all people said that polymerase 1 is the enzyme that removed the primer and refilled the gap also. When you look into the eukaryotic organism, RNase H1 is the enzyme that removes the primer, that remove all the primers except last primer. Now, why this RNase H1 can remove all the primer, why it is unable to remove the last primer? What is actually happening? Now all the students understand the concept very correctly. Whenever the most important polymerase, listen everyone, whenever the polymerase delta reaches the previous Okazaki fragment, the RNA primer by DNA synthesis create a RNA flap. Friends, what does it mean? Every student just try to understand. Just understand. Listen. So this is a primer, okay, and this is a DNA polymerase delta. What actually happening here? We all know very well the DNA polymerase delta will come with a higher speed. Very high speed it comes. So whenever it comes at a higher speed, actually whenever it reaches the primer, it has to stop. Whenever it reaches the last primer, the polymerase delta has to stop compulsory. But what actually happen, the DNA polymerase delta comes at a higher speed and it hits the primer. Whenever it hit the primer create a flap like structure like this, exactly like this. See, originally primer is like this, but upon hitting by the polymerase delta, it create a flap like this. You can observe a flap. You can see in the diagram as well, whenever the polymerase delta comes and hit this particular primer, you can see here it is going to create a flap like structure, right? So in order to remove this particular flap, we are using a separate enzyme. What is the name of that separate enzyme? We call it as FEN1, which means flap endonuclease 1 enzyme. What is that? Flap endonuclease 1 is the name of the enzyme that is involved in this particular process. There is no point of getting confused over there, and it is easy to understand also. So in the exam, what is the question they have asked? Which of the following enzyme is involved in removing the last primer in the eukaryotic DNA replication elongation process, which is the FEN1. Remaining all the primers removed by RNase H1. Please friends, don't get confused with this particular process. Then the next and another very important process that all the students has to focus on is the process of termination. Why this process of termination is very complicated actually? Why it is very complicated? What is the reason? The point is very clear friends. Why the termination process is very tougher is, let's quickly recall prokaryotic DNA replication termination, how easy that process is. If you quickly recall, this is the DNA of a prokaryotic organism. This is called as ORC. One replication fork is moving in this direction, another replication fork is moving in this direction. Correct na? So both fork meet at one particular point. That is only we can call it TER site, and this mediated by the TUS protein. So it is a very simple type of the termination, and moreover the DNA is completely circular DNA. Correct or not? But now all the students please try to understand, in case of a eukaryotic DNA replication, the process of the termination, every student listen, the process of termination is entirely different. Why? One of the reason is our DNA is the linear DNA. First, I will tell you what is the problem. First, I'll tell you what is the problem during the termination and how can we resolve this problem actually. Now, all the students try to understand. First, understand the problem. All the students listen, and I believe you remember this particular statement. I told to all of you, whether it is a prokaryotic organism or eukaryotic organism, the type of a DNA replication will be always bidirectional replication. There is no point of getting confused over there. Now, you see here friends, this is the origin. So one fork is moving in this direction and another fork is moving in this direction, which we generally call it as what? Bidirectional replication fork. No problem, no confusion at all, and it is very easy to understand also. Now, every student listen to me very carefully. Okay, understand my statements correctly here. What exactly you people has to understand, at the end you can see friends, here one template, sorry, here one primary is present, and at the same time here also one primary is present, which is shown by the red color. Now tell me what is happening. I have to remove that particular primers and I have to replace with the nucleotides. Okay, we have removed the primers. Okay, after removing the primer, the real problem will come. What is the real problem? Every student commonly know that if I want to add the nucleotide, what I require compulsory, that is important. If I want to add the nucleotide compulsory, what I require is the 3' hydroxyl group. Correct or not, friends? If I want to add the nucleotide compulsory, I require the 3' hydroxyl group. Now you only look into the picture, but where the primary is located, friends, all the students use your brain. The primary is located at the 5' direction. So I have removed the primers. Then what group is present there? Phosphate groups are present. Now you tell me, how can I add the nucleotides over there? How is it really possible? It is not really possible at all. Correct or not? Of course, yes, friends. It is not at all really possible. Now what is the solution here? What is the exact solution? Now the solution I will tell you first. Now since understand, all the students understand, the last primers are removed by the 5' to 3' exonuclease enzyme. That is right. But no DNA polymerase. Listen everyone. But no DNA polymerase can fill the resulting gaps. Why no DNA polymerase can fill the resulting gaps? What is the reason? Because there is a no 3' hydroxyl group is available. Because there is a no 3' hydroxyl group is available, which a nucleotide can be added. So now you tell me what is happening. So every round of the DNA replication, all the students try to understand, the DNA become shorter and shorter. Because we can't replace the nucleotides because there is a no 3' hydroxyl group is present. So each round of replication generates shorter and shorter DNA molecules. Friends, there is no point of getting confused over there. So now you see that DNA becomes shorter and shorter. Now you tell me, because of this what happened, friends? Most importantly, the cell senescence will occur, which means that the cells start becoming older very fast. The cell starts becoming older very faster. As a result, even sometimes it leads to the tumor formation also. Now tell me, now you all people understand what is the problem. What is the problem? We are unable to replace the gap at 5' end because there is no such DNA polymerase is existing. Now what is the solution? Now the solution for this specific problem is telomeres. The solution for this specific problem is what, friends? Telomeres and telomerase enzyme. You people can observe the diagram very clearly. What is telomeres? All the students try to understand me crystal clearly. Listen, all the students. Telomer. You can see that telomeres are what? Ends of linear chromosome. Okay, always important. They are the ends of the linear chromosome. What is the function? What is the exact function? One major function is it is protecting the end, no doubt about it. What, friends? It is protecting the end. At the same time, it is maintaining the length also. That's a crucial point, friends. It is protecting the end also, at the same time it is maintaining the length also. Protecting the end means what, friends? What exactly this meaning? Suppose this is the DNA. If it is not associated with the telomere, this entire DNA can undergo the degradation process very easily. What, friends? This entire DNA undergo the process of the degradation very easily. That's the reason telomere is important to protect the ends also. Telomere is important in terms of maintaining the length also. Now again there is a question which is asked in the exam. Okay, first telomere sequence, I want you to understand me very correctly, friends. The first telomere sequence identified from an organism called as a tetrahymena. Okay, the first organism identified from an organism called as tetrahymena. Why I am highlighting this is 99% they will raise the questions from the model organism every time. Model organism every time. That's why, my friends, it is super important. I want you people to remember it compulsory for the exam point of view. What tetrahymena? You know, 90% of the 90%. The telomere sequence in the tetrahymena and the human telomere sequence almost similar. So they can ask a direct question in the exam. Which of the following organism telomere sequence is homologous to the human? What is the question? Which of the following organism telomere sequence is homologous to the human? That is the tetrahymena. Please don't get confused. Again, there is a question which is asked in the exam. Which of the following nucleotides are rich of telomere? Are rich in telomere. So the telomeres are rich in the T also as well as the G also. You have to remember this general characteristic features compulsory for the exam point of view. Okay. Now all the students, please try to understand. This is what exactly we can call the telomere repeat sequence. You can see, right? So what it generally contain? Mostly it contain T and G sequence. Friends, never ever get confused with the point. Now I want to tell you one very important statement. The telomere always form a overhang whenever it is attaching. I'll explain you very clearly what is this particular overhang, how it will attach us. Now let's try to understand. Now every student understand what is telomere. What is telomere? The telomeres are present at the ends of the linear chromosome and the sequence is T and G. There is no doubt you got a correct idea. Now let's try to understand what is telomerase enzyme and what is the significance. What is the significance of this particular telomerase enzyme? All the students listen. The eukaryotic cells use telomerase to maintain the integrity of the DNA telomere. Okay. Now first I'm gonna tell you what is the telomerase enzyme is made up of. This enzyme made up of two components. One is called as telomerase RNA and second point it is called as telomerase reverse transcriptase enzyme. So simple, friends, no point of getting confused here. Telomerase enzyme contain two components. One is called as the telomerase RNA and another one is called as the telomerase reverse transcriptase enzyme. Now let's try to understand. So I said telomerase RNA. How long it is? It is very small. It is just 150 nucleotides. That's all. No point of confusion at all. Majorly it contains cytosine and adenine. Majorly it contains cytosine and adenine in this 150 nucleotide sequences. All the students please look into the picture correctly. This is the telomerase enzyme. What the telomerase enzyme contain? It contain RNA template. This RNA template is made up of 150 nucleotides. In this 150 nucleotides, majorly it contains cytosine as well as the adenine also. And what else it contain? Apart from this, it is also contain an important enzyme called as reverse transcriptase enzyme. There is no point of getting confused here. Now you understand how does the telomerase enzyme look like. It is crystal clear. This is the enzyme and this is the telomerase RNA. No confusions at all. Now first let's try to understand what is our real problem. I hope you can see that this is our real problem. Correct or not? What is our problem? We have removed the primers which are at the 5' end. Now we are unable to replace that particular gap. Correct or not? This is our problem. Now you friends see, observe what the telomerase enzyme is doing first. It is very interesting enzyme, I can say that. Listen, everyone understand the concept. First, listen all of you. This telomerase enzyme extend 3' end of the telomere. Let me repeat my statement to all of you. Actually, the problem is here. But what the telomerase enzyme is doing, friends, telomerase enzyme is extending the 3' end. Now you can ask me, sir, what is the use of extending the 3' end? Actually, the problem is here. That is a good point, valid question. The reason, whenever the telomerase enzyme extended the 3' end, whatever the end it is extended, that can act like a template. That can act like what? That can act like a template. Now telomerase come into the picture. Now telomerase having what, friends? Telomerase having RNA template. Yes or no? I believe all the students is getting my points very correctly. Now the telomerase enzyme come into the picture. Telomerase enzyme having what template? RNA template. Correct or not? Telomerase enzyme having, this is the telomerase enzyme, it is having the RNA template. Correct na? And we also studied that it is having what enzyme activity? Reverse transcriptase enzyme activity. So by using this RNA as the template and with the help of reverse transcriptase enzyme, you know, it will synthesize the DNA strand. By that it can fill the gap, no doubt about it. But all the students observe very correctly. Even though it repaired the problem, see, entire problem got resolved. But what is the new problem? New problem is that the telomerase enzyme always form a overhang all the time, all the time. You people can observe this picture, friends, very clearly. You will get an idea in this picture. Can you see this? See, friends, all the students understand this is the DNA. Up to here DNA is there, up to here. So but you see this is, what about this entire part? This entire part we generally call it as overhang. So what I am trying to explain to all of you is telomerase enzyme, listen all the students, telomerase enzyme always attaches to the DNA in such a way that it always form a overhang structure. Anyway, I am explaining you again. The people who did not understand, ask me again and understand the concept very clearly. What is our ultimate problem here? Our ultimate problem here is that, one second. So our ultimate problem here, I have to replace this particular gap. So what first telomerase enzyme is doing what? It is extending the 3' end. Why it is extending the 3' end? Whatever the end that we extended, friends, it generally act like a template. Whatever the end it extended, it generally act like what? Template. That point is super clear. Now this is the enzyme. What enzyme? Telomerase enzyme. Telomerase enzyme having what kind of primer? RNA primer it is having. Correct na? And it is also having reverse transcriptase activity. So by using RNA template and reverse transcriptase activity, it can start synthesizing the DNA. No problem at all. But what is the problem? You can see that it resolved our first problem. But what is the new problem? It always attaches to the DNA in such a way that it can form a overhang. What, friends? It always attaches to the DNA in such a way that it can form a overhang. So overhang is also one of the reason. Overhang is also one of the reason which causes cell to become aged very faster. That's the reason they may also raise a question in the exam. Which of the following DNA replicative enzyme involved in the cell aging? Direct question you can expect in the exam. Which of the following enzyme involved in the cell aging? All the students remember which is the telomerase enzyme. There is no point of getting confused over there. So this is about what? Eukaryotic DNA replication. Okay. Every student please confirm me. Is it clear, every one of you, replication? Anyone, any doubt, kindly ask me without having a confusion. Please resolve your doubts. So we just completed the first part, that is the DNA replication. The speciality of the molecular biology is, friends, compared to the replication, transcription is very difficult. Compared to the transcription, translation is utmost difficult. Now compared to the translation, gene regulation is super difficult. So that's the reason we have to understand this entire unit from very basic level to the very advanced level. Then only we people can answer this kind of questions. All right. So right now whatever we have studied is the replication. Now I believe all the students remember a point. What is the ultimate goal of the DNA molecule? The ultimate goal of the DNA molecule is synthesizing the protein. Correct or not? Of course, yes. The ultimate goal of the DNA is what? Synthesizing the protein. Correct, na? But we all people studied very clearly that the DNA never ever involved in the synthesis of protein directly. Instead of involving in the synthesis of protein, what it is actually doing? It is actually synthesizing an intermediate molecule. What is that intermediate molecule? We call it as the RNA. So synthesis of RNA from the DNA, we call it as what, friends? Transcription. So how can I give the simplest definition? DNA mediated. What is that? DNA mediated RNA synthesis. This is what exactly we call it as what? Transcription. There is no point of getting confused over there. Easy to understand also. All right. Now again, most of the students will get a confusion that, suppose example, this is a DNA molecule. In order to synthesize the RNA molecule, most of the students will think that we require entire DNA molecule to synthesize this particular RNA. No, not at all. We do not require the entire DNA. We just require a small piece of a DNA. We also call it as a small segment of the DNA. We require a small piece, or we require a small segment of the DNA. That small segment is sufficient to give rise to this particular RNA. Always remember that particular statement. And I believe all the students remember, in case of a prokaryotic organism where the transcription happens, the transcription happens in the cytoplasm. This is also 2000, I mean to say last month, this also we got the question. They have asked the question about the reverse transcription. In the upcoming slides, you can solve that question on your own. Straightforward, so simple question they have asked. Now, that is called as, so transcription is happening in the cytoplasm in case of prokaryotes, and in case of eukaryotes, the transcription is generally taking place in the nucleus. No point of getting confused over there. And all the students remember my statements, all of you. Transcription is the stage where the gene expression starts. I am always using one word, friends. Starts, I am saying, not ending. Okay. Gene expression starts from RNA level. And question is very clear. Where maximum gene expression takes place, friends? Maximum gene expression takes place at the translation level, because we are synthesizing the protein. That's why. So translation is the area where the maximum gene expression takes place. Transcription is the point where the gene expression just starts. There is no point of getting confused over there. Right. So transcription is the first step in the gene expression. Very important statement, friends. What is the major enzyme that is involved here? We're gonna read very depth here. Correct, na? Correct. Morning shift reverse transcriptase enzyme. Correct, Swati. We will solve it. Okay. RNA polymerase enzyme. Correct, na? So we will discuss that question very elaboratively. They just asked the tools that are required for the reverse transcriptase enzyme. Whether it require primer, whether it require a DNA primer, whether it require a RNA primer, whether it require a DNTPs, whether it require RNTPs, and whether it require the polymerase. Very easy. You people only will answer in the upcoming slides. Now listen. Transcription is the first step in the gene expression. Very important. What is the enzyme required here? RNA polymerase is the most important enzyme. Now all the students understand. This is what do we call, friends? We called it as a DNA dependent. That is an important statement because entire information is coming from the DNA only, right? That's why we can call it DNA dependent RNA polymerase. Why? Because the process of the transcription called as what? DNA dependent RNA synthesis. What do we call it as? DNA dependent RNA synthesis. There is no point of getting confused over there, and it is easy to understand. All RNA molecules except the RNA genome, you know, right? Every organism having the DNA as their genetic material, so they can synthesize the RNA. But there is an exception in the viruses, because viruses are having RNA as their genetic material, so they are going to give rise to the DNA. Correct na? And I told to all of you, whenever the transcription is happening, we don't require the entire DNA, we just require a small piece or a small fragment of the DNA. So listen all the students. During transcription, understand the point, an enzyme system converts the genetic information in a segment, only a segment. Segment means a small piece of a double-stranded DNA into an RNA. Now, you all people has to get one valid doubt. What is the doubt you all people has to get? Sir, DNA is a very big molecule. Then how can you confirm which fragment or which segment we have to consider? Why we are considering only this segment? Why can't we consider this segment? Why can't we consider this segment? The answer is very simple. It depends on what kind of a protein it is coding for. It depends on what kind of a protein it is coding for. That specific segment of the DNA only we will take in order to synthesize this RNA molecule. There is no point of getting confused over there, and it is easy to understand also. Okay, the ultimate goal of the transcription is what? Synthesizing the RNAs. So of course, if the protein synthesis wants to take place, friends, so we require every single RNA. We require mRNA, we require tRNA, we require rRNA. Again, there is a question in the exam, which of the following is the most abundant type of the RNA in the animal kingdom? Which of the following is the most abundant RNA in the animal kingdom? Which is undoubtedly rRNA. Why? Because rRNA is the one having a high stability and a long lifespan. Long lifespan and high stability also. Whereas, example I am telling you, friends, if you take the tRNA can withstand only seconds to minutes, that's all. Seconds to minutes. And if you look into the mRNA, it can withstand minutes to hours. But if you look into the ribose rRNA, it can withstand up to few generations. It can withstand up to fewer generations. That's the reason we can always say that rRNA is the most abundant RNA in the protein synthesis or in the animal kingdom. Of course, every RNA is very important. What is the ultimate function of the mRNA? As the name itself indicating, friends, mRNA is going to bring maximum amount of the information present in the DNA. What kind of information? The information that is required to synthesize the protein. Correct, na? Correct. Now, what is the tRNA? tRNA, as the name itself indicating, transferred. In simple words, tRNA sometimes we also call it as adapter RNA. What it is actually adapting? It is adapting the amino acid. So because without amino acid, protein synthesis cannot happen. So that tRNA only brings that particular amino acid. That's why we can call it adapter RNA. And the most important one, ribosomal RNA. Why ribosomal RNA is very important for the synthesis of the protein, friends? The point is super clear. It is generally involved, or it contains an enzyme called as a peptidyl transferase enzyme. The ribosome only contain peptidyl transferase enzyme. We widely call it as, I will tell you in the next slide, catalytic RNA. We also call it as ribozyme. What do we call it as? Ribozyme. We can call it catalytic RNA, we can call it ribosome, or we can call it peptidyl transferase enzyme. This is the one which are involved in the peptide synthesis or the protein synthesis. That's the reason rRNA is the most abundant type of the RNA. Alright, is it clear everyone? In the next class, we will read elaboratively about the prokaryotic transcription, then eukaryotes. It takes a lot of time. Okay, it's a very big lessons. Okay, clear everyone? Anyone having any doubt, you people can ask. Tomorrow also same time, 9:30 to 11:45, we'll complete the both units. Okay, we'll continue the both units. Clear everyone? Anyone having any doubts, you can ask. Requesting everyone compulsory revise the concept, don't neglect it. Clear? Alright, if anyone having any doubts, you can ask me. Alright, thank you all. Please revise it once again without fail. Thank you.